Okay, thank you so much. Can everyone hear me? Okay. Well, first, I just want to extend my thanks to Sophion for inviting me here. I had a really great time at their meeting in San Francisco. That's why I'm based out of right now, and they definitely showed us a good time. And really excited to tell you a bit about my work at UCSF. I'd also like to I'd also like to give a shout out to Derek, who he's my old boss in Montreal, and he was so gracious to get to share a 12-year-old photo of me in the lab. So thanks for that, Derek. I'm not going to have a photo of you in this talk, unfortunately. Okay, so I'll start with saying or introducing my talk by saying that action in a noisy environment, acting in the world, is very computationally demanding. Um, for example, if biking to the lab in San Francisco, there's a lot of obstacles that I have to look out for while also maintaining my direction, remembering where go to work, and partly so there's a lot of there's a lot of noisy stimuli that I have to filter out, and the way the brain does this, or at least partly, is through neuromodulator chemicals like acetylcholine and dopamine. This acts to silence the less salient, or the stimuli that you'd like to be less salient, and increase the saliency of stimuli that are more important to your behavior at that moment. So in the brain, computation in neuronal circuits requires integration of the inputs from multiple sources. So these are various inputs into this neuron here. And the neuron then decides whether to fire an action potential or not. And that's kind of the basis of computation in neuronal circuits. So neuromodulators can either act on the postsynaptic side over here, or the presynaptic side. On the postsynaptic side, generally exerts action through effects on various voltage-gated ion channels. And this has a generalized effect over multiple inputs. So this is kind of like controlling the gain on a stereo. This kind of turns down the volume on all inputs coming impinging on a given neuron. So pro is that it's strong control over inputs, but it's non-specific. Neuromodulators can also act on the presynaptic side, and that has the benefit of being able to regulate one input and while leaving another intact. So it's kind of like changing the EQ settings on a stereo. The problem is that due to complicated interactions between calcium in the intracellular or in the presynaptic terminal, this usually results in the response to subsequent inputs overriding that filtering effect. So there's you have input specific or input specificity with presynaptic neuromodulation, but there's this high pass filtering effect. Can you have a volume knob on a single input without changing the temporal dynamics of that input? So previous work in the lab investigated this question in layer 5 cortex in prefrontal cortex, and found that excitatory inputs there, even in the same terminal, can be regulated in multiple ways. First is kind of the canonical way, the traditional way. So this is an example of a GABA B receptor experiment. So GABA B agonist, baclofen, reduces the amplitude of the first synaptic response, but the relative strength of the second response increases even after stimulate, or even after the drug has been applied. So it can be said this is a decrease in amplitude, but increase in paired pulse ratio, as opposed to dopamine. So D1 receptors, the agonist at this terminal, causes decrease in amplitude without causing any effect on paired pulse ratio. This can be better visualized over longer trains of stimuli with multiple responses, and at the end of a train of EPSCs after GABA B receptor activation, there's very little suppression. This is what I mean by high pass filtering. So basically, there's no there's no suppression by the end of a longer train. Whereas with D1 receptor modulation, there's consistent suppression. So this is kind of like this volume knob over the entirety of the ongoing response to this neuron. So that gives you your kind of volume knob on a specific input. So this is just a better representation of kind of this filtering effect. So with baclofen, this dashed line is a scaled response. It is kind of measure the information transfer. So canonical modulation, like that by GABA B receptors, it's not effective at suppressing for long. So there's this kind of elevation relative to the baseline condition at the end of the stimulation. Whereas, so that's the EQ again. Whereas gain modulation by dopamine receptors suppresses input regardless of frequency or duration, which is the volume knob. So this allows gain control with input specificity. So this is possible, but what's the mechanism? How is this happening? So we found that both pathways still converge on calcium channels. This is the interesting thing. So how they differ is how they're actually affecting the channels themselves at a kind of biophysical level. So with GABA B receptor activation, the open probability of these channels remains unchanged relative to the previous action potential, but there's decreased flux per action potential. So that allows residual calcium to still accumulate, which causes a relative increase in the next response. Whereas with dopamine receptor stimulation, what happens is it causes these presynaptic calcium channels to shift into kind of reluctant gating mode. So there'll be occasional failures during each AP. So no residual calcium will accumulate, and therefore there's no PPR. So this is kind of the net change, no change in PPR. So the filtering stays the same. So the functional signature so far of D1 regulation is that there's this kind of selective effect on open probability, has this distinct gating mode shift. And it requires GS coupled signaling or protein kinase A. That's a crucial distinction, whereas GABA B receptors don't require those things. So this is a kind of hard transition to me, knowing what I know now. This sounds like an RGK protein. What is an RGK protein? So RGK GTPases, they're a part of this RAS superfamily of monomeric G proteins, but they don't really do G protein GTPase things. So there's 4 members. Hence their name, but they're really well known over the past 20, 25 years to prefer to inhibit calcium channels. And they have very, very strong effect on inhibition of particularly high voltage activated calcium channels. And they came onto our radar with this paper from Stephen Marx's group, where they found this protein, RGK protein RAD, regulates cardiac calcium channels and actually underlies the protein kinase A-driven fight or flight response, driven by the beta-adrenergic receptor system. So this is an example of a CAV1.2 kind of canonical cardiac calcium channel, co-expressed with RAD, becomes incredibly sensitive to PKA signaling. So this is with forskolin. So the pathway is as such. So basically, there's a stressful stimulus. Adrenaline binds to beta receptors. This drives intracellular protein kinase A signaling, which phosphorylates not the alpha subunit, but actually this RAD subunit. This is the PKA sensitive element in the system. And at least in the heart, RAD is bound at baseline to the calcium channel complex. The phosphorylation event then drives RAD away, thus kind of revealing the latent calcium channels on the surface of these of cardiac myocytes. And then this is in part what drives the heart rate increase, underlying the fight or flight response. So this was really interesting to us because this RAD molecule, protein kinase A sensitive, and it also has this kind of a unique effect on CAV1.2 open probability in a similar way that we would predict it would be happening at presynaptic terminals. So, the kind of logical extension is this happening in neurons? So RAD doesn't exist in neurons, or not really, REM2 is the kind of homologue of, or the isoform that's expressed much more in neuronal tissue and highly expressed in cortical neurons, which is where most of our experiments are. So could there be a similar system, like in the heart, at presynaptic terminals involving REM2 and dopamine receptors instead of the cardiac mechanism? So kind of a lengthy intro. The outline of the talk is, so is REM2 underlying this dopaminergic regulation at terminals and synaptic release in the brain? And then what are the determinants at a molecular level of REM2 regulation of presynaptic calcium channels, so these CAV2 molecules? Okay, so getting into some data. So how do we investigate REM2 in prefrontal cortex? So luckily, we didn't have to generate a new mouse line. This a floxed conditional knockout mouse was already made by Suzanne Paradis at Brandeis University in the States. So the experiment here is using an AAV approach in stereotaxic surgery, injecting a Cre to knock out the protein, to knock out REM2, as well as a Cre-dependent channel rhodopsin in the contralateral hemisphere of prefrontal cortex, and then recording in the other side, basically, recording from those neurons. And then we can use LED blue light to stimulate the D1 containing afferents onto those neurons. So what do we see? So in the wild type, this is non-REM2 floxed mice. This is after SKF, by the way, the D1 agonist. So there's a decent reduction, similar to what previous people in the lab have seen, and no change in paired pulse ratio. And the REM2 knockout in the same experiment after SKF had a significantly attenuated response to D1 activation. So this was really exciting to see. And the really interesting thing was that kind of basal measures of release probability at these same synapses, like PPR before SKF, so just baseline PPR, and then coefficient of variance, which is proportional to release probability. These were unchanged. So that suggested to us that REM2 might actually not be associating with CAV channels at baseline, and that it might need to be pushed to interact with the presynaptic CAV. Okay, so this is cool. So I say at multiple synapses. I didn't have time to show each experiment that I've done, but it's not just at that one synapse in the PFC. It's at other excitatory inputs to it. But really, I'm interested in the mechanism. And just to remind you, this is actually opposite to what you'd expect from RAD and cardiac CAVs. Remember that when the GPCR is activated, the RAD in the heart dissociates from the CAV. I'm saying that the REM2 would have to associate. So how does that work? How does D1 signaling drive REM2 interaction with CAV2? So what is different between these two situations? So for that we turn to trying to reconstitute this signaling complex in a heterologous system. So we chose to express the whole thing in HEC293 cells. So we have the CAV subunit, it's beta and alpha-2 delta, as well as REM2 and the dopamine receptor. So I'm using a slow ramp protocol, doing patch clamp electrophysiology, manual ephys, and recording these slow ramp protocols. So this kind of reveals kind of the whole kind of activation window of these channels. So get a nice ramp current there without REM2. And with REM2, there's a significant attenuation. So this kind of agrees with previous literature suggesting that REM2 does, when REM2 is co-expressed, at least together, that it does inhibit CAV2. But what happens when you drive D1 signaling? So my D1 agonist here, dihydrexidine, this just worked better for us in HEC cells. So after DHX application onto these cells, expressing the dopamine receptor, I get a nice time-dependent reduction in the overall barium current. Here, the summary data are as follows. So there's the largest effect, or the only significant effect, is when REM2 is co-expressed with the CAV2 and the rest of the accessory subunits. And then importantly, this is the kind of the before the VHAV activation before and after. There's no change. So this is important. So there's no change in voltage dependence, which is what we'd expect, or what we expect given our synaptic data. Some more different sort of evidence here. So we wanted to look at, so the idea here is that REM2 has to be by the membrane to be, logically has to be by the membrane, closer to the membrane to be inhibiting calcium channels. So we wanted to see if we could pick that up with immunostaining. So here, this is just, we're applying vehicle or the agonist onto hex cells expressing our complex, and then recording the membrane kind of associativity, the kind of proportion of REM2 that's by the membrane with and without agonist. And we see that with agonist, it's actually closer to the membrane, or there's a sharper peak there. So this is kind of again, like a morphological correlate of this functional inhibition that we see. And then the other cool thing is, so this is a different experiment, but same thing follows with, as before, the DHX kind of sharpens this membrane association, but it's pKa sensitive. So if you have a protein kinase A inhibitor in the bath, this process doesn't happen. So that's a nice kind of validation of that. But how does pKa control these interactions? Again, to this molecular idea. So this is a lot of letters. This is the sequence alignment of REM2 in 3 species in human and rodent alongside RAD. So first thing I'll note is that they're quite structurally homologous. They're pretty similar, especially at these putative sites, protein kinase A sites, these serines here. And I'll just draw your attention to the serines because the Marks group in their initial paper identified that there are these 2 serines in the C-tail, in C-terminal domain, that are actually really crucial for this regulation by protein kinase A. And if you mutated them, it made it insensitive to protein kinase A stimulation or beta adrenergic regulation. So we were kind of zeroed in on those serines there. So REM2 and RAD, and then I threw in GEM in here as well. These residues that are homologous in the sequence, potentially, again, this is a kind of theoretically at kind of different places when it's actually in the 3D structure in the protein. So to test that, basically just took a sledgehammer approach. I just wanted to mutate all of the serines in the C-tail, if the C-tail is still important for REM2, and see what happens to regulation by dopamine receptors. So this is this 5SA construct that we made. Wild type, this is the kind of wild type regulation with just wild type REM2 after DHX, and with all 5 serines in the C-tail mutated to alanine, so it's phospho-null construct, it's insensitive to regulation by D1. It's like, okay, that's nice to see that the principle still holds that these serines are still important. But what about these ones that are important in the cardiac system? So again, this is super preliminary. This is just literally from last week. I got an N-1 for you. So the 2SA, if you mutate just these ones, apparently there's still dramatic regulation. So that's exciting because it shows that there is this unique aspect to REM2. So we're kind of zooming in on this motif here that doesn't exist and has 1 or 2 serines in that region in only REM2. And that's what's coming up next. And with that, that's all I'm going to show you today. So I'll just wrap up with conclusions. So I've shown you a bit about that presynaptic REM2 does underlie dopaminergic gain modulation at central synapses. There are REM2 specific structural features that may explain its behavior at synapses, and that this kind of REM2 dependent gain control increases computational repertoire of the brain by allowing this kind of input-specific control over the volume without doing any filtering or changing the filtering. So I'd like to acknowledge people in my lab, particularly Kevin. He's been really supportive, and Sam, who's a really great tech who helps me with a lot of the TC work, and then the funding sources. And I'd be happy to take questions.
Thank you for your great talk. So we now have the time for questions. Any questions?
Okay, thank you. Great talk. So I have two quick questions. Is there any beta 7 specificity? You expressed the beta 3, but there are many different types of beta 7.
Yeah, so we've tried our kind of workhorse beta is beta 3. It's most highly expressed in cortical pyramidal neurons, which is where we're studying. Tried beta 4, it still works with beta 4, at least in HEC cells. So yeah, and we've also tried Cav 2.1 because yeah, it's kind of at most at least excitatory terminals, it's a mixture. Mature terminals, it's a mixture of N type. So Cav 2.2 and Cav 2.1. We tried both. It seems like it's pretty promiscuous in terms of beta and at least those two alphas. Yeah.
Oh, okay. Okay. Another question is that when the receptor is activated, okay, G beta gamma also could inhibit the current.
G beta gamma?
Yes, G beta gamma of the G protein.
Yes.
So did you exclude those kind of possibilities also?
Yeah, so this whole thing goes away if you block protein kinase A alone. So in that case, so we're not blocking the dissociation of the subunits from the dopamine receptor, but it's only the protein kinase A activated by the alpha subunit that seems to be important, at least for this regulation at synapses. In HEC cells, we don't have that. It seemed, so we have some data. I didn't show you the data, the protein kinase A inhibitor data in the heterologous assay, but that seems to be effective as well.
Yeah, Ryan, great talk. Fantastic talk, actually. I really enjoyed it. I probably missed this. You probably said this, but in terms of REM2 and RAD, it sounds like they're fine-tuning this calcium, presynaptic calcium regulation, or in the heart, it's a different kind of thing, but regulating calcium channels. Are those two proteins part of a big family of proteins that are all kind of structurally and evolutionally related? Is that right?
Yeah, that's what I kind of just glanced over on that one slide.
It looks like structurally there's a lot of similarity, right?
Yeah, it is like a very kind of, part of this traditional GTPase. It has like a GTP binding site and all that. Some groups think that the GTP binding itself is important for its regulation of CAV channels, but it seems like they're more well known, and they're the only subfamily of these RAS-related small GTPases that have any effect on calcium channel function. So they're kind of like a kind of weird branch off of this bigger family.
So they kind of block the ability or not the ability, they fall off or they block the ability of kinases to work, right?
Yes.
But do they do other things? Is that it, or is there other?
Well, no. I mean, the thing is, they're like this dynamic regulation. That's kind of the new, and that's what this Stephen Marx's group kind of really opened up, and we hope this is going to be the examination or investigation of it or report of it in the brain. But like they're around, like they do other things too. Like they're I think in various tissues, because they're expressed in peripheral nervous system too. I think they're a kind of way to kind of tune the kind of basal level of calcium activity at the membrane without having to take the calcium channels off. But yeah, because they provide really strong inhibition. But so just one more comment. The other group who works on REM2, Suzanne Paradis, her big interest is postsynaptic REM2, and it actually acts as a really potent inhibitor of CAM kinase 2 itself. So it kind of acts as a brake. So it's totally independent of any sort of calcium channel activity, or at least she hasn't investigated that. There might be some cool postsynaptic effect on calcium channels there, but remains to be seen.
Super interesting. Thanks.
So any other questions? We have 1 more minute. So may I ask you a question? So as the small GTPase is working in this model, so I am wondering if the switching by the gap or GAF will work on this control or modulation of this transmission.
Oh, like the other members
Yes,
of
yeah.
the RGK. Yeah. Yeah. So they could be for sure, because like there's a big literature about how each one of them inhibits each high voltage activated calcium channel, CAV1, CAV2, equally and just very strongly when coexposed. So if an RGK is there at the actual site of the calcium channel, it will probably inhibit it, at least if it's in the kind of, at least if it's not, regulated in the wrong way by protein kinase A or other kinases or something. So it's not really known, but potentially, yeah.
Other questions? So if there's no question, so yeah, it's time to switch to the another. So thank you very much.